Hello everybody. In this video clip I want to illustrate how we can analyze a DNA agarose gel and when we are looking, for example, for an unknown DNA fragment. So here we've got our agarose gel, and we have a sample here which we want to analyze. And in particular, we want to analyze this band here, this labeled X, the red band. We want to know what fragment size this band X is. And that is a very, very common problem that we encounter in any molecular biology lab. So we've run our sample on an agarose gel, and together with our sample, we also used a marker which gives us well-defined DNA fragments. And these fragments here are, for example, in base pairs. So base pairs, because it's DNA. So our first DNA fragment in this marker would be 10,000 base pairs, and it goes all the way down to 50 base pairs. And we let this marker run together with our sample so that it gives us sort of a standard. So we can now actually construct a standard curve and then try to figure out how big this red band actually is. And before we get started, we can already see that the size of this fragment is probably somewhere, or definitely somewhere, between 1,000 base pairs and 2,000 base pairs, maybe somewhere in the middle, so around perhaps, well, 1,300 to 1,500 base pairs or something like that. But we want to do it a little bit more precise, and therefore we need to do a proper analysis so that we can exactly say how big this DNA fragment is. So how are we going to do that? Well, what we are going to do is we simply measure the distance of each of these marker bands from the start. And to do that, we just simply take a ruler, which we apply, and the start, that would be here, the start of the DNA. Well, so that would be the start. And all we need to do is really, we need to measure how far these samples have actually traveled from the start. So we see that the first band, the 10,000 band, the DNA fragment band, has probably traveled around, well, I would say 0.75 centimeters. So let me put this in. So that would be 0.75 centimeters. And the second band, the 5,000 DNA fragment, the size has traveled around, well, that's 1.75. 1.75. So these are the data, and here I have typed them all in. We would, of course, we would also do that for our unknown band here. And again, we measure it with the ruler, and it's probably 3.65 or something like that. So I've now did this table for all the DNA fragments down to 50, and I measured the distance with the ruler from the start, including the unknown band here. So here are actually the data that of the DNA fragments and the distance from the start. So let's just simply draw a graph to see whether this is a nice linear relationship between the DNA fragment and the distance traveled from the start. And our independent variable, that is the variable that we have control over. that would be here our x value, whereas the dependent variable, which depends on the DNA fragment, that would be our y value, just to make sure that we understand what we plot. So we would plot DNA fragment on the x-axis and the distance from the start on the y-axis. So let's just simply highlight them all, and in Excel we go to Insert, we do a scatter plot like this one, and we get a nice linear, actually no, we don't get a linear relationship. Let me just quickly label the axes. Here is on the x-axis is the fragment size. That is in base pairs. And on the y-axis, that's the distance traveled from the start point. And that is in centimeters. And what we see is that this certainly is not a linear relationship. So we cannot really apply anything that we have learned about standard curves. We could sort of say, okay, we've got 3.65, that would be roughly here. So that would be here. So where would this be? But we can't really draw a straight line, and we can't do any interpolation with that because it is not a straight line. So how do we actually deal with that? We cannot directly plot the data DNA fragment size versus the distance traveled. Now it turns out actually that there is a relationship between the DNA fragment size and the distance from the start, and this relationship is not just a direct linear relationship. It turns out that is actually the logarithm to the base 10, or any base, log of DNA fragment. And let me make this a little bit bigger. Make it look nice, good. So instead of plotting DNA fragment size, we need to plot the logarithm of the DNA fragment. And Excel can do that quite nicely. All we need to do is we need to transform our DNA fragment size into the log DNA fragment size. So this is equals log, and we use the log 10 here, log 10 of our DNA fragment, and we get our fragments here. Let me just make this again a little bit bigger and center it, and we do this calculation for all our DNA fragments here. So the way I do that, I'm too lazy to type that in, so with the mouse I go to the right-hand bottom corner of the cell that I just calculated, and you see the black cross appearing. With the left mouse button clicked, I just simply drag it down and let go of the mouse, and I get my log 10 DNA fragments. And now let's take this away. And what we do now is we plot the log 10 fragments on the x-axis versus the distance from the start. So let's highlight our cells. Highlight them. No. left mouse button clicked, and then on a Windows keyboard it's the Control left mouse button clicked. So I can highlight two separate columns. Now I do again the insert. I go to charts and I choose a scatter plot. Make that a little bit larger. Move that around. And again, I've got on my y-axis, I've got the distance traveled, but on the x-axis now I have actually the log to the basis of 10 of the fragments. It's a little bit smaller so that we can see everything. Okay, so what we see is we have a really nice straight line which illustrates that we are not supposed to plot DNA fragment size, but log to the base of 10 of the DNA fragment size. And then we get a straight line. We now can calculate a trend line for that in Excel, and we know how to do that. So we will left mouse click on one of the data points so that we highlight them, then right mouse click, and we can add a trend line. So here this trend line, it looks pretty good, so there is a very strong correlation. We know it is a negative correlation, so we have a negative correlation, because the larger our x values, the smaller the y values, and that is indicative of a negative correlation. Just lost. So trend line. Now what I can do, and I showed you that in a previous video, I can display the equation on the plot on the chart, and I can also look at the R-square value, which gives us an indication how good the fit is. And I make this a little bit larger here so that we can easily determine things. So what we see is that we have an R-square value of 1, which means perfect correlation. So that's really nice. And we have our gradient for this standard curve. The gradient is negative 3.376, and the intercept for our standard curve is 14.235. So this is the gradient and the intercept of our straight line. And what we can do now is we can try and calculate what is the log of the DNA. What is the log of the fragment? Fragment X. So we know that fragment X has traveled 3.65 centimeters. So we could go over here and where we hit the standard curve, and we can go down here. So, but we need to be aware that this is not the fragment size X that we are reading here. We are reading actually the log of the fragment size. So we need to be aware of that. But we can also calculate the log of the fragment size. Remember, we have our equation here, y equals mx plus c. That's the equation for the straight line of the standard curve. And if we want to make x the subject of this equation, we just simply transform that. So we bring the c to the other side and then we divide by m. So we would get equals y minus the intercept divided by the gradient. And we can do that here. Let's just simply put this in. So for the log x fragment, we would get equals, let me make this a little bit bigger again, later, equals, we have 3.65, that's our y value, minus our intercept, that would be 14.235, and then we divide by the gradient, and the gradient is negative 3.376. So that's our gradient, and we would get, let me make this large now, we would get for our log fragment, and it's important to realize this is the log fragment x, we would get 3.14. So that's what we would get here. Now, how do we convert the log into non-log? We know that in order to convert a log, what we need to do is we need to eliminate the log, and we can do that because it's base of 10. We can do that by taking 10 to the power of this value, and that would give us our fragment x. We can do the calculation here. Equals 10 to the power of this one here. So we would get the fragment size for x of 1366 base pairs. Now, of course, we need to be a little bit careful because we've got uncertainties. If you look, for example, here, when we look at the band sizes, it would be very difficult to say that this is exactly here 3.05. It could be, quite a bit of things, a bit of distance. So we need to be a little bit careful here. And I think the best way to deal with would be to say that the fragment that we are looking here, the fragment is around probably 1400 base pairs. And that gives us enough room for any uncertainties. Does that answer make sense? Let's just simply go back to our gel. And previously we said we estimate it to be between 1000 and 2000, probably somewhere in the middle. And we just got 1366, well, 1400. So this band here, X, we are pretty confident that this is around 1400 base pairs. And this is actually how you would analyze a DNA sample. It's really, all you need to do is you measure the distance of your marker bands and the band that you are interested from the start, and the start is the beginning of the well. And then the big thing is that instead of plotting DNA fragment size versus the distance, you plot the logarithm of the DNA fragment size versus the distance. And then you get a nice, usually you get a straight line. Sometimes the endpoints are not exactly on a straight line. That has something to do with the fragment sizes moving a little bit slower or faster for extreme ends, but we are not too much worried about that. And from this straight standard curve, we then can determine the log, and that's important, the log of the unknown band. And we can convert this log then into the DNA fragment size. So I hope this makes sense, and thank you very much for watching.